welcome to this scientific lunchtime seminar by ECHA. Today, we are going to hear and see Professor Emma Schymanski of University of Luxembourg talking about environmental cheminformatics. That's about finding relevant pollutants in Europe and beyond. Our presenter is Professor Emma Schymanski. She heads the research group for environmental cheminformatics at the University of Luxembourg. She's both environmental engineer and chemist, and she has worked in the past for Golda Associates, for the Environmental Research Center in Leipzig and in Switzerland before she moved to Luxembourg. Her interest is in developing methods to identify unknown chemicals and their effect on human health. And she has a very impressive record of citations, publications, and a book. And I looked up her most cited paper, actually got up to date roughly 2,500 citations. So that's impressive. Emma is an advocate and advisor for fair and open science, and I'm sure she will going to explain to us what that stands for. And now I will hand over the floor to Emma. And Emma, I will make you presenter now, and then the floor is yours.
Thank you very much for the introduction, and thanks to all of you for tuning in. If I occasionally look away from my camera, it's because my slides are right next to me on a different screen. So I'd like to talk to you today about, as Jan mentioned, environmental cheminformatics, finding relevant pollutants in Europe and beyond. And yes, a big factor of this is also open and fair science. So I'll go into also a lot of the tools that we develop to help explore our contamination in Europe. My slides are actually available on Zenodo, so if you would like to download these to have references to read up later, I do cover a lot of material. You can either use this QR code here or just the DOI available. And I'm, as Jan said, I'm also happy to have any feedback of points during this talk. Okay. So let me first click on. All right. So what is environmental cheminformatics? And our primary measurement technique for this is non-target high-resolution mass spectrometry. Basically, we take a look at the environment. We have many different samples. Once we have this sample, we will interrogate it or measure it with high-resolution mass spectrometry. Then comes the cheminformatics, or the data interpretation. And out of this, we wish to identify the chemicals and their potentially toxic effects. And to do this, if any one of you is familiar with programming in R, these hex stickers are very common. We develop also and co-develop a wide range of community and open activities to support researchers in general in navigating this very complicated chemical space. So the vast chemical space of exposomics or environmental sciences, as I'm sure you're all aware, it's incredibly large. We can't actually enunciate all of the chemicals that are known. PubChem, one of the largest open databases we have, is 119 million. CAS is 219 million. But unfortunately, MassBank, which is one of our basically fingerprint databases of all the mass spectra of the chemicals we know, this is less than 0.01% of PubChem. So we have very little measured data for a lot of these chemicals. This is where the non-target high-resolution mass spectrometry comes in. So if we would do a classic targeted analysis, we've just got a very narrow view at a few tens to maybe hundreds of chemicals, depending on the reference standards available in-house. Then we have what we call suspect screening. This is where we look for chemicals that we think will be of concern. So we could say, for instance, a list of pharmaceuticals that are registered in Luxembourg. We would, obviously, if they're registered in Luxembourg, we would hope or suspect we would find these in the Luxembourg community. And I'm going to mention this a little bit later. We host the Norman Suspect List Exchange, which hosts various suspect lists. Originally, they were meant to be lists of, hundreds to maybe a couple of thousand chemicals. Over the years, this has grown and is already 127,000 chemicals. So you'll see our chemical space is extraordinarily complicated. Then finally comes in the non-target screening, where we just take a look at the breadth of the sample that we have. We record all of the masses there, and then afterwards try and identify what these masses or what chemicals these masses could belong to. This picture has been very carefully designed because whatever analytical technique and extraction technique we have, we know we will be missing a huge chunk of the chemical space that is a part of this as well. What we also do, in addition to the Norman Suspect List Exchange, we also have this PubChem Lite for Exposomics collection. This is how we're trying to break down this chemical space into a more informative subset to also help scientists and chemists come to grips with this huge chemical space. I'll go more into this a little bit later. So non-target high-resolution mass spectrometry, this is basically one technique. You can do a whole lot of different sampling campaigns with this. This is just an overview figure from an article in 2017. It's hard to believe it's already so many years ago. We have various experimental setups. So either you can do laboratory experiments, you can do treatment experiments, or you can just do routine monitoring. Once we've performed sampling, classically, you would also start establishing a sample archive. These days, with the high-resolution mass spectrometry, after analysis, we can also do what's called a digital archive, so that over time we can actually build a snapshot of what's happening in these samples without actually having to archive the entire sample. And then afterwards comes the fun for the informatics side. So we've got the data pre-processing, then you've got some form of prioritization, and then identification. And Jan requested a few case studies rather than a lot of details today, so I won't go into great depth in the methods. In the next slides, I'll just show you a few different examples of how we can prioritize and then interpret in different contexts. So one of our classic and I think one of the nicest examples, and I'll come back to what instigated this campaign a little bit later, is we now have the real-time monitoring of the Rhine River. This was obviously done while I was at EOWG. And already this actually started before I joined EOWG. This is a daily collection of samples. So this snippet is from 2014 to 2016. As you'll see on the axis here at the bottom. And what we have here on the y-axis is actually the concentrations. And you can see we can detect unknown chemicals due to their standout patterns. So you'll see here in the top one, PPA, this was unknown before we found the signal. You can see the first peak discovered in 2014 is actually 0.6 tons of this chemical coming through the Rhine. And later in the same year, another 0.1 ton came out. Basically, with our data processing, there was an intensity threshold that was hit with this chemical, which instigated investigations. Then they looked further up the Rhine where this was coming from, got enough sample intensity to elucidate the structure, and actually followed this up with the company to see. What we can also do, and one of our problems is you can also have just a background signal that we don't know what it is until all of a sudden for 6 weeks this signal disappears. So this is what's happening in the second picture at the bottom here. You can see we always had a signal, and then all of a sudden there was a production break of 6 weeks. And this actually also triggered a reaction in the data processing. Was, oh, this signal's dropped out. We were actually able to track it back through the Rhine. And as you can see, in 2015, the company actually instigated a mitigation measure. It didn't completely get rid of the signal, but it did dramatically reduce the number of tons coming into the Rhine. And so with this, you can see we can actually use non-target mass spectrometry, even if we don't know the identity of the chemical, to find what's going on and understand what's going on in the environment. Another really nice example we had from Luxembourg. This was my postdoc Randolph and my PhD student at the time, Adeline. We had a look at the pharmaceuticals, as I'd already mentioned, and we saw a really surprising trend. So I don't know if you're all aware, Luxembourg is actually, obviously we're a very small country. Our population doubles during the day and halves again at night. So we have a lot of cross-border workers coming into the country. And what happened in 2020, of course, everyone was in home office because of COVID. Our pharmaceutical concentrations in the surface waters dropped dramatically. So if you have a look at the pictures here, we've got in the purple was the 2019 concentrations, and in the green, systematically lower for almost every pharmaceutical, there's actually the 2020 concentrations. And again, here we saw some obvious trends. Nicotine, of course, if you halve the number of smokers in the country, you'll get a dramatic drop of the signal of nicotine. And here, this was quite interesting. Iohexol, which was one of our highest intensity pharmaceuticals in 2019, actually dropped out completely in 2020. And this was because the hospitals were so busy treating COVID patients that they weren't doing any of the cancer diagnostics anymore. And this was is a chemical that's commonly used in diagnosis. We've also had a look at the pesticides in Luxembourg. We actually do a lot of work following up on this with the transformation of not just pesticides, but chemicals in general. So here we've got one example, and this is where you can really use the retention time through the mass spectrometer and then the occurrence in different months to see whether transformation products are actually associated with the chemical you think it is, or maybe with a related one. So what you can see here is the middle two peaks. In terms of retention time, they make sense according to the transformation, but the peak you can see for both of them is blue. They're occurring in September, whereas the primary product was occurring in July, whereas the one at the bottom, it's also got a green peak. Retention time makes sense according to the transformation product, so it's much more likely that this bottom transformation product actually belongs to the parent at the top. And these other two signals in the middle, they're obviously genuine signals, but they might belong actually to a different parent pesticide. I'm also a member of the Norman Network, which is a network of environmental laboratories in Europe and beyond. And as part of this, we also have a large number of resources that we maintain. And this is the digital sample freezing platform, which is actually primarily Nicky Forrest's work. We actually provide the chemical information that he then uses to screen. So you can basically, with a drop-down menu, pick a chemical, and then use the mass, but also the fragmentation information where we have it to extract where this chemical is occurring. And you can basically build up a picture, a spatial picture of the distribution of this chemical throughout Europe. And this is basically, it's a live interaction with our entire substance collection. So by now it's about 120,000 chemicals that you can just build up this picture. The nice thing with this digital sample freezing platform is this also allows the retrospective screening. So this is a subset of REACH chemicals in the Black Sea. And what you can see over time, we can see here the first third, we have biota samples. You can see a few chemicals are coming out in the biota. A lot of them are coming in the water. So you can see the top third here is really occurring in the water. And then you can see the chemicals occurring in the sediment are actually a different group of chemicals. So over time, we can really start to see which chemicals are actually occurring in the different matrices to help focus our efforts as well. Stepping back to Luxembourg, here are our latest non-target screening results. So we actually have had surface water samples since, I established my group here in 2017. So from 2019, we started receiving samples. By now, we've built up a few different years. One specialty of Luxembourg or our routine monitoring is we have 4 locations that are the same every year. So these are marked with the square, and then otherwise it rotates in a 3-year cycle in different locations. So we don't have a consistent data set for all locations for all of the time, but we have quite a big picture over time. Actually, our water management agency does the targeted monitoring, and we basically do complementary non-target screening. And over time, based on the chemicals that we detect, we can then classify them, whether we have known uses for these chemicals, whether they're potentially associated with any disorders, diseases. As you can see, as not particularly unexpected, we have a large proportion of pharmaceuticals. These are generally higher concentration. And then we have about 15% agrochemicals. And then we can build some trends over time. So here you see just from 2019 to 2022, but also the fluctuation during the year. So what you can see here, obviously the pharmaceuticals are dominating. So we still see this dramatic drop in COVID times, which actually surprisingly has not recovered as much as we expected. We have also noted that Luxembourg has become much friendlier to home office since COVID happened. So maybe this is a result of this. You can see the pesticides have dropped a little, but not much. And what we also see is when you look at the interannual percentage, the pesticide spraying is definitely obvious in the spring and summer months because we get an increase in the pesticides in these times and go down again. And we do also notice in the pharmaceuticals, we get the trends you would expect with antidepressants increasing in winter and this kind of thing. What we can also do, this is looking at a more local subset of Luxembourg. This is the corner very close to where we are. The university is just around here, actually. We can then do a map of the different industries that are around. This is close to Petange. This is the very small river Shears. And then we also have a wastewater treatment plant that was just upstream of our sampling point. And if we look at the information we have from industry versus the wastewater treatment plant, it was unfortunately extremely clear that pretty much all of our signals are coming from the wastewater treatment plant because these identifications are pretty much tracking with what we see in the wastewater. So again, this kind of points to the pharmaceuticals and the human consumption as being one of the drivers in this river rather than the industry. What we could do based on this is we basically took a look at the 41 frequently occurring chemicals that were not yet monitored. And again, we had to look at the classifications. As you will not be surprised by now, quite a few of them are pharmaceuticals, or in the blue, they're transformation products. We also had a few industrial chemicals that we flagged, and the organophosphate flame retardants, which are beginning to be concerning actually throughout Europe as well. And Luxembourg seems to be no exception there. So how do we support all of this? We do a lot of open source cheminformatics developments. As I've kind of mentioned already, the number of chemicals we're faced with is extremely high. So we're trying to help also get the information out there, but also help people navigate this. And one of this is, of course, the mass bank, the spectral library. This is actually the measured fingerprint of the chemical. This is how we identify them with the greatest confidence. I'm just going to highlight here, we have a nice arsenic compound that was one of our first editions during Anjana's master thesis. This was in 2019. She actually, after this, we became, I think, the 4th largest contributor to mass bank. But we've really tried to fill in some of the gaps. This was based on the US EPA intact data set. We're also, as I've already mentioned, gathering the expert knowledge. This is the Norman Suspect List Exchange, and Heba is helping me out with this. This is the entire Norman database system. We have several different database modules. We're working primarily on the Suspect List Exchange. You can see Mass Bank is also here. And with the Suspect List Exchange, we're hosting different lists so that people can find these suspected chemicals a little bit easier. As I mentioned, it started in 2015 with very small lists. Now we're up to 121 different lists and 127,000 chemicals, 131,000 substances, because not all of them are actually discrete chemicals. And we're integrating a lot of this information into the Norman Suspect List Exchange and into PubChem as well. We published this in 2022, and as you can see from the author list, this was 97 co-authors. We didn't quite hit the 100 mark. We were very close, but you can see this is really a huge effort of a lot of different researchers providing information on a wide range of different chemicals. There's no way one single person would have managed this effort alone. And how does this work? We get a whole lot of different suspect lists from people. You can see some of them are for us, an informatics dream. They're basically everything annotated very easily. Some of them are beautifully formatted for people to read, but not so easy for computers to work on automatically. We standardize them to an extent. We upload them to the Norman Suspect List Exchange. We put them on Zenodo. Through Zenodo, we actually get this unique identifier, a DOI. What this also allows us to get over time is actually a look at the views and the downloads. And this has had actually an extraordinary impact on being able to track the lists of interest, because what we hadn't realized is our most popular list was actually a EU cosmetics list. We'd been busy uploading PFAS and food contact chemicals and all pharmaceuticals, pesticides. Meanwhile, our cosmetics list was almost 10 times as popular as any other list we had on the Suspect List Exchange. So this can also help us as researchers decide and see which are the actually most important parts of our work. Once we have it in the Suspect List Exchange, this is integrated into Sustat, which is then the substance database that feeds all of the other Norman database system. For example, the digital sample freezing platform. So this is where this drop-down menu comes from. It's basically integrated in the central database. What we also do, we don't just stop there. We don't put it just on Norman, but we integrate it with the US EPA Comtox chemicals dashboard. And also a lot of content goes into PubChem as well, not just the chemical list, but also actually the measured data and the transformation reactions as well, so that people can actually extract these again in a different way. We actually connect a lot of this knowledge through METFRAG. So METFRAG was at the beginning just an in silico fragmenter, take a mass spectrum and take some candidates and try and match the candidates the best way to the spectrum. In the meantime, this is actually for us summarizing all of the information from the resources I've just shown you. So what we'll basically do, if we have a mass that we're interested in, we can query a different database for the candidates. So for this example, we have the mass of nicotine. Querying Comtox, we have about 140 candidates, but we use various scoring terms, and you can see quite a few, or a few candidates at the beginning here have quite high scores, and then we have a really long tail. But our grand challenge is, of course, that our chemical space is too big. So I already mentioned we have CAS is now 219 million chemicals, PubChem 119 million, ChemSpider is around 129, and even the CompTox dashboard is at 1.2 million. So if we would query, for instance, this massive nicotine with PubChem, and if we're seeing nicotine in our samples, it's most likely to be nicotine, we will end up with a few candidates with very high information content, and then we have this extremely long tail with very low information content, and this is giving us, in terms of our computational methods, prohibitive runtimes. So we will basically, with a 20-minute sample, we would be running this for 2 days to run our normal identification system on it. And I've shown this this slide for many years, and people are like, yeah, okay, it's 3,000, it's a big number, it's okay, numbers are fine. When I actually explained this problem to a musician, this is Jamie Pereira, he actually tried to put this problem to music for us so people can actually appreciate it. And I hope this is going to work. Please let me know if it doesn't. It starts very soft, but it should get quite loud. So if it gets too loud, I'm sorry. I'm going to narrate it as it goes. So what we've got here, starting at 1950, these are the number of patents for different chemicals. We've got DDT at the top here, PFOS, PFHXS, PFOA, and then here's just the total of the patents. On the axis here, we've actually got the CAS numbers registered per year going up to 20 million. So by the end of this, you will see we've got 20 million coming up. The ticks are actually the years going by. 1962, Rachel Carson wrote Silent Spring noticing that the rivers are running dead because of DDT. Here you can see the CAS numbers are beginning to increase, so we're only getting a couple of hundred added per year. Rachel Carson was already concerned about the number of new chemicals per year in her book. We've got a couple of industrial disasters coming up, so this was an industrial disaster in Italy. And you can see the patents are beginning to increase, so we've got a little bit of a yellow coloring happening here. The PFOS is obviously not increasing so much yet. And you can see the CAS numbers are beginning to trend upwards a little bit. Here was yet another industrial disaster. Here was the Sandoz chemical spill that actually stimulated the Rhine monitoring that I explained a few slides ago. In the 90s, we started the DuPont scandal in West Virginia with PFOS. I'm going to turn this down a bit. I hope it goes down. And you can see this ran for about 10 years before there was any legal action that started. And you can see the patent numbers are only just beginning to increase, and so are the CAS numbers. So you can see by the year 2000, we've only just begun a restriction of DDT. And these are the chemical numbers we're faced with. And they just keep increasing. So I kind of started my non-target career in about 2010. Already the numbers were looking pretty bad. But they're getting even worse. So late 2009, we finally got a restriction for PFOS. But you can see the CAS numbers keep increasing. Some years they drop a little bit, but that's still 10 million being added a year. And this is kind of where we're at in 2021. So you can actually download this film if you would like afterwards. It's pretty shocking. I really like the way the music explains. This is basically, oh, you didn't hear the music? I'm sorry. If you didn't hear the music, on the next slide, you'll basically, if you look at this QR code, you'll be able to download the soundtrack. Sorry, we didn't have a chance to check this with Jan. It's a really intense soundtrack, but this basically shows you really nicely in this one picture the amount of information that we're faced with, but also what you as ECHA probably are also faced with, is this huge number of chemicals. How do we come to any form of decision with this? And I've already mentioned it. We've got this huge chemical space is too big. So one thing we were looking at tackling is how can we get rid of these candidates with low information content and really reduce the prohibitive runtime and focus on the chemicals for which we have enough information to act. This has been really important to empower non-target screening. So what we did is we actually had these are Evan, Jeff, and Paul from PubChem. We basically started trying to subset PubChem. So the number of chemicals in PubChem with information is about 70 million. So even though the entire database is 119 million, they have, let's just say, non-trivial information for about half of them, so 70 million. But if we break this down even further, here you can see in the yellow, we've got various categories of relevance for environmental investigations. So we've got the agrochemicals, we've got drug and medication information, food additives, identification methods available, pharmacology, biochemistry information, safety, hazard information, toxicity, and of course, use in manufacturing information. It's really important for us. But what's more is if they're listed in this category, there are also subcategories that you can see. But if we have a listing, it also means we have some form of data available in PubChem that helps us interpret this, this entry. So here you can see this one has an entry in EU pesticides data. And if we would detect it, we can actually see it's not approved. And so then not only do we have a detection that we know is registered for some reason, but we also know, okay, we should be concerned about this because it's actually not approved for use anymore. We also have, for the exposomics point of view, we have the associated disorders and diseases information. This is not necessarily that the chemical causes disease. It can also be that the chemical is a therapeutic. So what you see here, again, nicotine is one of our favorite examples, but this is actually therapeutic in some neurodegenerative diseases. But what you can see is we also get the evidence and the references for people to look up further. For the metabolomics people, we have these interactions and pathways if you really want to do the full-on interpretation of the metabolomics pathways and which effects this chemical could have on other pathways. What we've also done for PubChem Lite, it was originally just a CSV file that was a download. We've now produced also a web interface so that people can actually browse the collection. You can download the latest file on Zenodo, so it is completely open and accessible. But through the web interface, every chemical has just a little summary page. With this, we put the basic information, the annotation hits, the number of references, the number of patents, the mass, and of course the predicted X log P, which is very good for our retention time prediction. But what we also do is provide a little snapshot of the information that's available, collision cross-section information, and I'm going to go into this in a little bit more detail. Also the literature and the patent stripe that shows the trend of this chemical over time. And you'll see from that previous video, we see a very similar trend. Usually it starts in a green zone, and then towards the most recent years, we get a real increase in the number of patents and literature mentions as well. So what we managed to achieve with PubChem Lite is, again, here we're back to the example of nicotine. Instead of having now 3,000 candidates with PubChem Lite, we have close to 30 candidates. And we also have these informative scores for people to sort. So we have the patent and the PubMed count and the annotation count as well. And we have the live checking of a mass spectral database as well. So we've created a much more efficient subset of PubChem for people to be able to use with non-target screening. We didn't just stop there, though. This is also integrated into a full workflow for non-target high-resolution mass spectrometry. So we work very closely, with Rick Helmus, and he does this entire workflow that I showed in the image before. So the pre-treatment, the prioritization, the full annotation workflow. I won't go into the details, but it's a very comprehensive tool set. What we do with this, and we still work on adding new functionality. Again, Jan didn't want too many details, so I didn't go into any of the informatics specifics with any of this. But what we are trying to do is, if you have your suspect in your non-target screening and you find these chemicals of interest, we will do a live collection and try to see if there are any related transformation product information, so that you can then do a second round of screening, screen for all of the related transformation products and or parents as well. And then gradually build a picture of where the different transformation products and where the different suspects could be, and see how they could possibly be related. So it was... thanks for sharing the video, Jan. So mentioning PFAS, and I mean, this was... TFA is being heavily discussed at the moment, so it was a quite nice example that we'd already set up for this slide. So we did create a little bit of a stir a couple of years ago when we mapped up the the new definitions of PFAS to PubChem. So I actually wanted to dedicate a few minutes of this talk just to going through what we tried to achieve here, and also show you how you can use some of the tools that we've developed to try and navigate the space, and hopefully see, yes, it's bad, but we can actually break it down into subsets that help people find the information that they need. And this relates also to the video because, yes, the numbers are shocking, but actually the numbers of PFAS that match the different masses we have in the environment, we often run out of PFAS candidates to explain some of the masses even. So, and this is a lesson learned in, are there really 6 million PFAS in PubChem? I submitted this talk for the webinar, and by the time I actually gave the talk, it was already up to 7 million. So these numbers are actually increasing by thousands per week. So we are literally, we're rebuilding this tree every week, and the numbers are going up. We're actually very, very close to hitting 8 million in this category by now. So what was the motivation for this? We took a look at the updated OECD PFAS definition. They came up with this nice figure in their report. And we basically took this figure and tried to scale this to PubChem and see how much of PubChem can we actually fit into these different figures. Because when I talked to Evan, he just looked, CF3, this is going to be several million in PubChem. And then when we mapped it up, yes, indeed it was. So let me just run you through the tree. So what we did is we, in the end, we had to expand it to PFAS and fluorinated compounds because you can't just restrict this to the PFAS. And as you can see, yes, the numbers are shocking. It's 22 million. So we have this organofluorine compound category. This matches with this entire category from the OECD. And then what we did is we have this OECD PFAS definition that matches with their updated guidance for the OECD PFAS. What we did was then we added the OECD PFAS and matched them to the salts and the mixtures, and this is where you got the extra million. You can see here it's about 6.8 million, here it's 7.9. So the problem actually gets worse if we map it through to the salts and the mixtures. What we also had to do was add this other diverse fluorinated compound section because we noticed a whole lot of very bizarre PFAS-like chemistry in PubChem that did not match any of the guidance provided by the OECD, but is obviously highly fluorinated content that we could potentially be worried about. And as you can see here, this includes things like this phosphor compound that is now also becoming quite concerning, even though it doesn't have a single carbon in it at all. So over 6 million OECD PFAS in PubChem, where is this all coming from? You can see we have around 700,000 with an isolated CF2 group. The vast majority of them, 6 million, are actually isolated CF3 groups. And this is because the CF3 is a very highly favored binding group in drug discovery. So a lot of PubChem is also virtual libraries from drug discovery. So this explains these rather large numbers. But even if we look at the molecules containing PFAS parts larger than CF2 or CF3, we still have only, in inverted commas, 227,000, which is still a huge number of molecules with long chain PFAS in them as well. But like I said, you can actually break down this quite quickly. So you can break this down by the functional groups. And for instance, if you're interested in PFAS connected to a ketone or an aldehyde group, you can break this down. And then if we're especially interested in the carboxylic acid, you can break this down further and then break it down by the part formula and find we actually only have 459 that have a C3F7 with a carboxylic acid group. You can then query these numbers in PubChem and actually sort them by different categories, relevance, mass, all sorts of things. And then you can actually see what data in PubChem matches this category. What we also did as part of the tree is gather together all of the different suspect lists that many researchers are compiling as well. So we have over 50 suspect lists from the CompTox dashboard. And again, if you expand in this arrow, you can get the whole listing. We have several lists from the Norman Suspect List Exchange. We have the data extraction that we did with Google on OntoChem from patents. This is quite large numbers. Not all of them are suitable for suspect screening. And we also have this really informative list from NIST. It looks quite small numbers in comparison to the others. But what NIST did is they actually enumerated some of the PFAS that are only available in mixtures, and with this list they actually added over 1,000 new chemicals into PubChem that weren't actually mentioned in PubChem yet. So this again shows the importance of actually sharing and integrating the data in open resources so that we can all find the data and help solve these rather challenging problems. Finally, what we did, which I hope will be of perhaps particular interest to the audience here, and a huge thanks to Andreas Buser from the Föhn in Switzerland for this interesting, I mean, we worked on this together, the interesting perspective of how to also make this tree useful for the regulatory decisions. And here we break down according to the major categories that are under discussion. And I'm going to take the example of PFHXS because this is actually a really nice one because he himself was faced with this discussion. He has the EU REACH wording and then he has the Stockholm Convention wording, and there was actually a very small difference in the definitions, and he was trying to assess, okay, what is the implication for Switzerland if I adopt this one or that one? And if you actually look at these little info bars here, we have the full text from the regulatory definitions actually integrated so that you can see upon which basis we've gathered the information. And the difference between these definitions is literally whether you define a C6F13S moiety or a C6F13S with 2 double bonded oxygens. And that is the major driver of the difference in the definitions. What we also did was put out the different category that actually lets people download just the substances that were different between the definitions, so you can actually see, is there actually an implication for our jurisdiction if we have these substances regulated or not? And again, it sounds quite large with 114, but if we break it down into just those for which we have extra information, you'll see we actually only have 14 for which we have any literature, use, safety, toxicity information. So again, you can go down to the breakdown and see, okay, which ones of these do we actually have any information to interpret the chemical? So again, all of these categories, if you click on the blue box, you could basically send the query to PubChem. So here, if we would get the 114 that are different, you can then browse what is the difference. So here you can see clearly the difference. We have no double bonded oxygen, so it's a 6-valent sulfur that is fully fluorinated. And what's more importantly is you can download this in a variety of formats and then browse these results. And with the download file, you actually get a lot of the data that is also displayed here to browse. So this is just a screenshot of the download file. I've kind of color-coded it a little bit so you can see what information is there. So we have in the red, we have the basic name, identifier, smiles, all of the formats that the cheminformaticians like to have. In the blue, I've highlighted a whole lot of the calculated properties. So for us, for mass spec, we like the X log P, but there's also heavy bond, donation and acceptor counts, complexity counts, polar area, the masses, of course. And this is basically, if you look in the PubChem record, this is exactly the data you see in the summary pages as well. So it's basically just in an export format for you. This is going further along in the download file, so there's really a lot of information hidden here. This is not so intuitive from the header, but here is the literature and the patent count. So the PCL is a consolidated literature count, and the GP is the Google patent. So this is a patent identifier count and the patent family count. And here you can see in the blue, we've actually got the annotation and the deposition categories. So you can see a lot of the concern with the PFAS is that they're just coming from patents or from vendor libraries. So you can see here the chemical vendors. But you can also see some of us are either it's curation efforts or they're being contributed by research and development. So that means it's coming from researchers who are detecting these or governmental organizations. And you can see here the researcher here, this was my group in Luxembourg. You can also see, for instance, patent scope. This is just coming from the patents. And here under the annotation hits, you can also see whether we have use in manufacturing information or the literature, or if it's just the classification category. What we also have is the date this was added to PubChem. So PubChem has only existed since 2004. So obviously there's no dates earlier than 2004. But what this does help you do is also pick out the chemicals that have been newly added if these are of particular interest, to see if there's anything coming from research that's very recently added. This is something that might be of particular interest. All right, and this gets back to what I was saying before with this. So these annotation hits, this is related to the PubChem table of contents. And that means if we have use and manufacturing information, you can actually go back and find whatever has been that information that has justified this categorization. So here we have all sorts of different information, and in some cases we even have production statistics as well. So because PubChem only existed since 2004, we noticed that we were having a little bit of trouble reconstructing the chemicals over time. And we'd also seen from that video that it was actually very useful to look at how the chemicals are trending over time. So this is actually the work that Dagny did with her visualization with the patents and the literature. And I'm just going to run you through a few examples. So we've got both the patents here. This is PFOS and literature. There is quite a lag time in the patents, so you'll notice that almost all of the patents end with a green, and that's because it takes about 2 years for the data to come through, and the literature is pretty much alive. It's updating every week. And if any of you have ever developed any informatics software, you'll know that lesson number 1 is always test random numbers, and in this case, here you'll see our classics, caffeine and nicotine, beautiful patterns. But here we just randomly did CID1 because that's Paul's favorite test. And I did 1, 2, 3, 4 because I just like the number 1, 2, 3, 4. And 1, 2, 3, 4 actually ended up being really, really interesting. So you can see the vast majority of the stripes have this pattern. We have green, and then towards the 2000s, we get a very high increase in the numbers. And for the patents, like I said, we kind of get a green bit at the end. You can see acetylcarnitine, this is CID1. This is a biological molecule. We don't get so many patents on this, so you can see the data increases, but it's kind of a bit patchy. And then for galopamil, we were like, okay, this is strange. There's a big peak in 2007, and then it just dies. That's a bit weird. And then when we looked at the literature, it got even stranger because you can see here 1, 2, 3, 4 galopamil actually peaked in the 80s and then went down to nothing. You can see all of the other ones had kind of the same pattern, kind of green and going towards red. When Dagny looked up on this, it turns out that galopamil was actually a pharmaceutical that was quite promising for a certain treatment. It went into clinical trials, had extremely bad side effects and was taken off the clinical trials later, which would explain why we had the peak in the literature, then the peak in the patents, and then absolute silence because it was obviously not going to be the chemical that solved, or the pharmaceutical that solved that issue. So this was a quite interesting take home for us because we're like, okay, the pattern is not just this general increase, but we can actually tell if chemicals have some kind of history over time. And what we did, this was also, we made a little R package. So while Daniel Zahn, he's working on PMT compounds, while he was talking at CTAC 2003, we actually did a live chemical stripe calculation. And he was mentioning, this was the phosphorus compound I mentioned before, but he also mentioned the tetrafluoroborate. We kind of did the stripe for tetrafluoroboric acid and we're like, okay, it's the same pattern. But then he mentioned that lithium tetrafluoroborate was actually the chemical of interest. And this was the only stripe we've seen actually go red and orange towards the end and not go back down to green. And this is a chemical associated with electric vehicle production. So we were like, okay, this is really, the patent numbers in China were going through the roof. So again, proof that we could see trends in the chemicals through the patents numbers. So what we did, this is Flavio, he was a student job on the side during his master's thesis. This is Jun Pang from our computer science, and Paul again from PubChem, who does the patent work. We were basically looking, can we then do a regional analysis of the patent information and pick chemicals that are up and coming in different regions? And I just wanted to give a little bit of perspective of where the regulation can really help. So just bear with me while I go through a few of these patents. So what you see here is we've got in the purple, the US patents, in red, Korean, green, Japan, orange is the European. The blue is China. So you can see these are generally coming up in the last few years. And then the gray is basically other. So we took a look at the agrochemicals and then the subset of the biocides, and you can see it's kind of the same pattern we've seen for everything. Starts off very low, and then all of a sudden we get hugely increasing numbers and then a sharp tailing off in the last couple of years. And then when we looked at bisphenols, we kind of see the same thing again, even a little bit more exaggerated. But then when we looked at the PCBs from the list on Comtox, you can see we see a completely different pattern. So we kind of see an increase, and then you can really see regulation did actually kick in with the PCBs. Yes, there's some patents coming in, but very little. So we have not seen the big peak in the PCBs that we did with a lot of the other chemicals that we're seeing. And when we looked at the PFOA list, so this is the initial PFOA list that the Stockholm Convention listed, we saw a very similar pattern, actually. We saw, you could see it kind of kicks up at the beginning, but then we also get this drop-off much earlier than for the general trend. But then when we looked at the updated PFOA lists, this was only updated a few years ago, you can see because they weren't regulated earlier, we actually had quite a few chemicals trending up towards the end. So this actually shows that people are actually following the regulations. Obviously, some people always try to get around them, but we are noticing some effects in the patent numbers. And again, we're seeing actually quite a large increase in China at the moment as well. So I know this is probably going to be quite an intense seminar, and there's so much to tell and not enough time to tell it. What you can do, again, the PubChem PFAS tree is just one example of what we're doing. We do have documentation. If you go to this little click out box, this will actually take you to quite a long documentation where we walk people through how to use the tree, what the different categories mean, how to navigate the information. We did also publish this year, last year as well. And we have a couple of, they're getting a little bit out of date, the interactive walkthrough and webinar from 22 and 23. And we also do have, for expanding beyond PFAS, we have the PubChem Light collection. It's about 400,000 chemicals, where we have also the information that's available and the trends of the stripes over time, just to see which chemicals are of concern and which ones are maybe not so concerning. So I hope I've been able to show with some case studies that non-target screening workflows are extremely powerful. So we're able to put into practice finding different chemicals and interpreting how relevant they are for different contexts. We do develop a wide range of open and fair cheminformatics methods to support non-target screening, try and present information in an easy way for people to digest, even though the numbers are actually shockingly large. But how can people break down and actually come up with useful conclusions for their studies? We think the patent and the literature data really holds a lot of potential, where obviously the stripes are just a very rough estimation. We're actually working still with the people who are extracting this information to try and refine these a little bit further. And I think we all know the number of chemicals is a huge challenge, but I think this is also where we have to help share the chemical information publicly so people can actually navigate this and come to their conclusions. So with this, I'd like to thank you all for listening. I hope this was some interesting inputs for you. A huge thanks to my fantastic team here. Again, you can download today's slides if you'd like to read up on any of the information. A huge thanks to all of our collaborators and our funding agencies for funding this effort. And you can see over the years, this has indeed been hundreds and hundreds of people who have contributed to this. And thank you again. I'm happy to take questions.
Thank you, Emma. That was yeah, extremely fascinating, dense indeed, but very, very interesting. And I bet you have a few more fans in our audience now, and we already have got one question. So yes, can you see Kaishu's question? Would you like to answer it?
Yes. How do I think our group and the EU agencies could help each other? I hope I've managed to show how we can. I mean, obviously, we do a lot of work with sharing the information as well. We've downloaded a lot of European data. We've integrated it into PubChem, and what I can say from our experience with Norman is, I mean, I think the download statistics we got for this EU cosmetics list, this was almost certainly driven by our integration in PubChem, because they literally have millions of users a month, and the Norman network is still too specialized. So within Europe, I think in the meantime, it's known. But this is how we've tried to work with multiple different agencies, because the more ways people can find the data and interact with it, actually, the more we can help people. So what we've tried to do, I actually didn't include the slide, I should have, the amount of PFAS data that we've been able to add from different researchers. So we've looked at what Christian Svina has been publishing on the transformation products on the Superfund sites near Rastatt. These structures were not in PubChem before, but we're actually able to put them there with the spectra so people can find them. And then we're working with Erin Baker. We've integrated the use information from Juliana Glüger. So I mean, if there's any way that we can somehow help, we're happy to do so. I have connections with a lot of the other databases by now. Yeah, it's basically starting a conversation and finding the win-win, and some of it is actually surprisingly easy to do. So our entire PubChem integration is automated. We add new data. Of course, we have a couple of manual steps. So we do a check of performance before we do any automatic commits. But we're updating this information constantly. So, and if you have ideas, extra data you need, I'm more than happy to pass on any knowledge I have.
Well, yes, indeed. Thank you very much for your answer. And please, audience, don't be shy. It's your chance now to have a live exchange on this fascinating topic. We had a look back in the past of chemicals. We saw how the patents and use evolves. We got a musical inspiration here. So any more questions in the audience? Or maybe Emma, you would like to add something or ask us? You feel free to ask us, you see.
I was going to say I didn't even mention it in my slides. I think the biggest challenge for all of us at the moment as well is also these UVCBs, right? So, I mean, most of what I've shown today has been on the discrete chemicals, but you know, the next challenge that we will be looking into is how can we actually map the constituents to these UVCB chemicals and try and find out how much these are contributing, because we definitely see peaks that are coming through as groups of chemicals that are almost certainly coming from technical mixtures as well. So we know the PFAS, there's probably quite a lot of PFAS from that, but also a whole lot of surfactant groups and others and that. So yeah.
Are we going to enjoy the silence, or are there any more questions in the audience? I don't. Is there any hand raised? I'm sorry. I may. The Fleur, please go ahead. I'm sorry, I missed it.
Yes, thank you. Thank you very much, Emma. It's simply very, very big, I think, what you've been presenting, and there's so many interesting things in there. Maybe a very ignorant question where many of us are trying to identify those substances that we should act on first in terms of further regulatory action. If we would like to use your information, and maybe we are already, or some of us are already digging into your information, where should we start?
Yeah, it's a good question, because a lot of people, when we did the PFAS tree, they're like, this is too many chemicals, you need to prioritize them for us. But everyone has different reasons for prioritization. And this has been one of our biggest challenges as well, because whenever we ask people how do they need to prioritize the chemicals, they were like, but you should prioritize them for us. It's like, but we don't know what your questions are. And I mean, this is why I wanted to spend the time and showing the download files and showing the information that's there, because in the end, what we've found the most useful is the information that's in PubChem, because you can get the download file. We have those categories. When we evaluated MetFrag, we definitely saw the literature and the patent values really help improve our performance. So the identification performance with PubChem Lite is close to 94% correct, whereas most of the methods just based on mass spectrometry, the best one out there is at about 30% correct, correctly estimated. So I know there are people that say we shouldn't use this extra data to score chemicals, but in terms of environmental outcomes, it is most likely that those with the highest patent counts, the highest literature counts, are actually the ones that we're going to be seeing in the environment. And this is actually why we've focused so much on the information. So the use in manufacturing information, if there's information there, this at least helps us interpret it. And we've found the same even when we're working, I didn't show any of our exposomics work, but when we're working on the disease outcomes, it's the same. If you can have some sort of interpretation, where is this chemical coming from? Is it in household use? This is something we can act on. If we just have, okay, it's an interesting chemical, it matches the mass, but we don't have any information, then there's nothing for us to do. So from that point of view, yes, we're using the literature count, the patent count, the information categories. Obviously, if we're looking for agrochemicals, we'll prioritize agrochemical information, but for other ones, it might be the use in manufacturing or toxicity is a really tricky category. We've found this actually never improved any of the rankings, because it only, the way we integrate it, it only says whether a toxicity assessment has been made. It doesn't say whether it's toxic, and whether it's toxic, of course, depends on the context as well. So this one we've really struggled with. And the reason we've looked at both the literature and the patents is the patents helps us capture a lot of those industrial chemicals that are not necessarily in the literature. So we've actually found the coupling of those have been, even though the stripes I showed, mostly they look the same, there are actually cases where only one or the other are following. And then, like I've said, you know, prioritizing, we are trying to work with the researchers that we know are doing cutting edge PFAS work. So we're like, especially Erin Baker and her lab, because they have very top analytical setup, the Christian Svina as well. And we're strategically working with them to get their information into PubChem. So if there are new PFAS coming from research organizations, we're also prioritizing those because they're actually seeing them in samples. You know, they're measuring the crocodiles, they're measuring the soil, they're measuring the animals, and they're seeing those chemicals. And then this is how we're trying to say, okay, these are the ones we should be focusing on because holy crap, they're already in the animals. And then we should be worried. So yeah, I know it's a lot of information, but it can actually be broken down quite quickly. And again, I'm happy to also spend time helping people. What I've actually done in this documentation, it's about 30 pages long, but every time I get an inquiry, if I can, I try to add a little bit of a snippet on how to do that query to answer that question. So, you know, how do I find the PFAS that have been measured on LC-MS that are either an agrochemical or a pharmaceutical? And in the end, it's only a couple of hundred. So you can actually break it down quite quickly. So the numbers look scary, but depending on your question, you can actually break it down very quickly and then use those scores to sort out 2 or 3 or 4 that you're most interested in. So. Yeah.
Super, thank you.
Yeah, don't be scared by the 7 million. I mean, we are scared by it, but it's, yeah, it's hopefully manageable somehow.
Dear audience, don't be scared by the numbers. And Pia is not scared. Please go ahead.
I was just wondering if you know if your data has been used to prioritize substances under the Water Framework Directive for the priority substance list, or identifying these substances for the watch list. Thank you.
Yes, in both directions. So out of the Norman Suspect List Exchange, we have several priority lists in there. So Norman was also involved in some of those priority lists. And I don't know if you remember it from the slide on the Suspect List Exchange, but we actually have a number of citations. So I think 230 of our lists have been cited now. And through that, we can see which lists are actually being used in different articles. We know the number of people that are using it that aren't citing the list is also quite large. So the citations are a small fraction of what people are using. But all I can say is since we've done this PubChem PFAS work, the citations for that article are going through the roof. I have a citation alert on all of those, and people really are using it. And okay, some of them are probably just saying the numbers are bad. But actually other researchers are actually using that to prioritize. The cool thing we found about the patent stripes even is because we presented that at CTAC, someone has actually already published a paper using the patent stripes to interpret his work before we'd actually published the patent stripes ourselves. And this is kind of why we're doing this. It's like, okay, get the information out there as soon as possible because people are actually using it very quickly and using this to find other substances or in other contexts. I mean, he was working on, I mean, it was in the Baltic Sea, and they had time trends from the 80s, and they were looking at, okay, which chemicals matched the trends that we could see in the patents. And this was really interesting. So, okay. Yes, I don't know if that...
So did I understand correctly that all of your data is in the Norman database? Or did I somehow not understand it correctly?
No, yes and no. We host the Suspect List Exchange, so we have a lot of data in there. The entire PubChem PFAS tree is not in the Norman database because it's 7 million chemicals and it's not set up for that. So again, we do a prioritization, which are the most relevant for Norman, and through Norman, this is through the suspect list.
Okay, yeah, thanks for
so people provide the suspect list they're interested in, we upload it, and then it's integrated within the system, and this is our way of prioritizing what people are most interested in. So, and yes, this SUSTAT, so the entire database, the 127,000, this is what Norman then uses for its prioritization efforts that's led by Valeria Duglio.
Yeah. Okay. Thank you.
So, yeah, we could integrate everything, but it would be too much. I mean, it's at some point, really, these numbers are extraordinarily difficult to build computational resources on. And it's like we're already struggling with 120,000. And I mean, building PubChem Lite every Friday, this takes 4 hours. So it's not computationally trivial to do.
Yeah, I can imagine.
Yeah. So
Okay.
yeah, we try to find a good balance.
Thank you.
I don't see any more hands raised. So, well, then I think that's it for today. We pretty much spent 1 hour on your topic, Emma. And yeah, thank you so much for this great presentation, for sharing your slides. And we will later on be able to watch the video with soundtrack, I guess. And dear audience and dear Emma, please, you will receive a feedback email. Please give us feedback also, yeah, about who else you might want to see maybe during a lunchtime seminar, or what kind of topics you want to be covered. So that's all your ideas are very welcome. And with that, I would like to close today's lunchtime seminar. Again, thank you so much, Emma, and it was great to have you here around. And hopefully, yeah, maybe I'm sure we will meet one day or another somewhere.
Hopefully. Thank you very much all for listening, and thank you, Jan, for being a great host as well. Really appreciate it.
Thank you, and have a good day, everyone. Bye.
Thank you. Goodbye.